Hi, my name is Ofer Elhanani. I'm a staff scientist in the lab of Dr. Liat Keren at the Weizmann Institute of Science, Israel. In the lab, we focus on research questions related to interactions between tumor cells and their microenvironment, including immune cells and stromal cells. And one of the questions that is of interest to us is how are these interactions related to the genomics of tumors? So when we first started to look of different ways to analyze genomics of tumors, it was first important to us to have a protocol that would be easy to integrate into our lab, since at the time we did not have a lot of experience with next generation sequencing technologies with our own hands. We also wanted to have a convenient analysis pipeline so we could get results quickly. And we had a list of genes that we wanted to ensure that would be covered in the gene panel that we analyzed. We also wanted to analyze some large-scale genomic metrics such as HRD and TMB. Regarding the HRD, and we actually had a chance of since some of the samples that we analyzed were also analyzed by competitors' kits and technologies, we actually were able to compare for a small set of samples the results both for the specific mutations analysis and HRD, for example, and results look very consistent with competitors, so we completely trust the data that we get. In order to implement the protocol of library preparation and enrichment and later on sequencing and analysis in our lab, we had to have a very clear protocol, and we wish to have very good guidance. We needed to buy new equipment for the lab, for example, in order to integrate these protocols into the lab. And with that, Karim from OncoDEEP was very, very helpful. We met more than several times. All my questions were addressed. I was able to very easily understand which equipment do I need to get in the lab, understand the protocol, which was explained to me stage by stage, and eventually successfully implementing it into our lab protocols. We ended up very quickly analyzing 120 tumor samples within a time frame of about 3 or 4 months. We had all the results that we wanted for. We got from zero experience to having 120 samples analyzed within 3 or 4 months. Analysis pipeline, the OncoKDM, is very well suited, I think, for the clinic, with a lot of information about clinical trials and suggested drugs and things like that. Whereas in my group, where we are a research group, we are more interested in the research side of things, and we had slightly different needs. So the OncoKDM database that includes a very elaborate information about different mutations and clinical significance. Uploading of data from the sequencing to OncoKDM was amazingly easy. It went very smoothly. I would expect while transferring such large amounts of data that there would be crashes and things like that. But no, everything went very smoothly and even quite fast. I got a very clear explanation on how to do it. I would definitely recommend it. My major factor is the service. I got and quick answer to any question that I got. I got a very, very close guidance and training to do something from zero experience to getting the results. And I think that is very important, and therefore I would definitely recommend it.